Here I've launched SeqMatNGen, and I'll click on New Assembly, and then in the Variant Analysis and Resequencing tab, I'll go down to the PacBio area and choose Whole Genome. I'm going to be using a reference sequence downloaded from the DNASTAR database, so I just click on Download Genome Package and choose the Drosophila melanogaster genome. For the sequence file, I already happen to have it open in my File Explorer, so I will just drag and drop it into the Input Sequences screen. The read technology is PacBio Hi-Fi, and the experiment setup is going to be single sample. Assembly options I'll just leave at the default values, but for analysis options, I need to click the diploid phased button that triggers this workflow. And the gender is male. Project name, I'll just call this It's recommending that I can run this assembly on this computer, so I'll just click the button and assembly begins. Once assembly is complete, I want to open the results in GenVision Pro. So to do that, I'll press the upper button, Analyze and Compare Variants in Multiple Samples. When the assembly opens in GenVision Pro, the first thing that you'll see is the genome view, which shows phasing blocks as alternating blocks of blue and green. However, we can't really see any detail in this particular view, so we want to look at it on a chromosome level. I'll double click on chromosome 4 so we can open that in the analysis view. And I'll just go to about the center of the chromosome so that we can take a look at what we can see in this view. So this view has a few different things that we can see depending on the zoom level. This is the default zoom level that allows us to see everything on a base pair level. And if I scroll a little bit, look for red or bright blue characters, and those will indicate SNPs or other types of variants. I can also go here to the search bar and search for variants this way. This was indicated as a variant, but it looks like everything in this row is a C. So let's scroll down and see what we can find. So note that this is showing in green, that's indicating one of the alleles. And now we can see that the other allele is the one that has the variant. That's shown in a darker shade of green. So you can see one of the alleles is in the bright green and one is in the sort of olive green. This is controlled by going to the tracks panel, clicking on alignment, and then you can see here, this is where you can see what the color scheme is. So this is an even block, so color A is the bright green, color B is the olive green, and you can change that here if you like. Looking further down in the tracks, this is a bifurcated type of variance track that's showing that the variant is in the one allele but is absent in the other one. Now let's see what happens when we zoom out of this view. Now we can see the individual features and hover over them to get more information if we like, and we can tell which variants are associated with a particular feature. For instance, in the first segment of this one-cut feature, the first variant is found only in one of the parents and not in the other. If we scroll out even more and then use this vertical scroll, we can see where one of the phase blocks transitions to the next phase block. So there we've got a blue phase block, and here's where we can see it has transitioned to the next phase block, which is shown in green. To show all of the variants for this project, I'll go to Variants, Show Table with Default Filter. That's just one of several options. By clicking on this gear icon and choosing Settings, then Variant Info, you can see there are 3 different items here that have phase in the name. I'm going to select all of those and move them into the display column box. These are the columns that will be displayed, and click OK. And I can just double click any row in this variance column and it will open that chromosome in the analysis view and snap to that position. And I can expand this by just showing the one view, and then we can scroll down and you can see that there is no variant in this first allele, and there's the variant in the other allele.